So hello everyone, I'm Meghan Pinezich. I'm a PhD student in Gordana Bunyak-Novakovic's lab, which is part of the Snoeck Hub at Columbia. Thank you very much to the organizers for the opportunity to present on our recently published study entitled Xenogeneic Cross-Circulation for Extracorporeal Recovery of Injured Human Lungs. So the motivation for our work is the shortage of donor lungs. Currently, about 80% of donor lungs are not suitable for transplant at the time of donation and are instead discarded. So our goal is to take these lungs that are initially deemed unsuitable for transplant and recover them over time so that they can be transplanted into a patient in need. So currently, ex vivo lung perfusion, or EVLP, represents one really promising approach towards achieving this. EVLP is able to take marginal quality lungs, these are lungs that just fail to meet the transplant criteria, and recirculate a hypertonic perfusate to flush out metabolic waste products and reduce edema. And despite significant progress in the EVLP field over the past 20 years or so, the most pressing limitation of EVLP remains the limited duration of support, which is about 6 hours maximum before lung function starts to decline. And this 6-hour window doesn't really allow sufficient time for meaningful intervention or regeneration of injured donor lungs. So thinking about the limitations of EVLP, our group began to realize that the missing ingredients limiting ex vivo support systems were really the function of other organs. Our bodies have evolved to have complex tissues and organs that work together in systems to maintain homeostasis, not alone in an isolated circuit, right? So we realized that in theory, if we could just add other key organ functions to an ex vivo circuit, like renal clearance, liver metabolism, and pancreatic energy management, we could potentially provide a homeostatic physiologic environment and possibly extend the duration and improve the quality of extracorporeal support. So this realization is what led us to the idea for cross-circulation. In cross-circulation, we include all of these other organs and provide their respective functions by actually incorporating the recipient patient into the circuit. So the patient essentially acts as a bioreactor for the donor organ that he or she will receive. So the injured donor lung is connected to the vasculature, connected via the vasculature to the recipient and is perfused with recipient blood. In our preclinical configuration, we have used a swine model as our recipient. And over the past several years, we have worked to develop this system and demonstrate that that lungs can be supported and recovered using this platform. So we first reported on our use of cross-circulation in 2017 when we demonstrated that we could maintain lung function outside the body for up to 36 hours and recover swine lungs with ischemic injury. We then demonstrated that cross-circulation enabled regeneration of swine lungs with severe gastric aspiration injury, again across 36 hours. And then last year, we extended the duration of extracorporeal support out to 4 days, or about 100 hours, this time using a conscious swine host. So what these 3 studies have in common is that they were all done in swine lungs connected to a swine recipient or swine host. But we were and we are really interested in bringing this work closer to clinical translation. So we next asked ourselves if cross-circulation could similarly be used to recover injured human lungs. And the answer to that question is yes, but how we arrived at that conclusion is what I am going to tell you about today. So we obtained 6 human lungs, which were declined for transplantation for a variety of reasons. And I'd like to draw your attention to the fact that one of these lungs was previously connected to a conventional EVLP circuit for 6 hours, but failed to recover in that time. So all of these lungs were transported to our cross-circulation operating room and connected to swine recipients via the vasculature, marking the start of xenogeneic cross-circulation. So here you can see our operating room general setup with the anesthetized recipient draped here on the left, the donor lung supported in a temperature and humidity regulated dome. And then on the right, you can see kind of the overall setup of our OR with an organ cart with real-time online controls and readouts. So this time-lapse video shows recovery of human lungs supported on xenogeneic cross-circulation for 24 hours. And you can see in the video when we take the dome off, it's a little bit hard to see because the dome fogs up. But you'll be able to see that the consolidated regions of lung, particularly in the lower lobes, were successfully recruited and ventilate normally after the first few hours. Additionally, mean changes in functional parameters across 24 hours confirmed that xeno cross-circulation supported recovery of respiratory function. For example, lungs showed improvement in P to F ratio, that is the ability of the lungs to oxygenate the blood, which increased steadily throughout cross-circulation. Similarly, dynamic lung compliance improved consistently. And although the lungs gained weight in the first 6 hours, shown here, this is indicative of slight edema. This was largely resolved by 24 hours and lungs returned to baseline weight. So as was shown in the time-lapse video, the gross appearance of human lungs at initiation of xenogeneic cross-circulation revealed really extensive consolidation, edema, and hemorrhage. However, throughout cross-circulation, as revealed by gross appearance of the lungs as well as radiography, there was progressive clearance of this regional consolidation and increased radiolucency by the end of the experiment. Similarly, bronchoscopy initially revealed mild secretions and airway inflammation. However, this was also largely resolved by 12 and 24 hours. The histological analysis confirmed maintenance of conducting airway structures, including intact pseudostratified epithelium and segmental and subsegmental bronchi. Parenchymal edema and cellular derangement were also largely resolved by 24 hours. Looking at scanning electron microscopy revealed the initially perturbed alveolar architecture that you can see here due to hemorrhage and parenchymal ultrastructure were restored by 24 hours. And transmission electron microscopy showed the retention of the blood-gas interface throughout 24 hours of support. So after 24 hours, we saw successful lower lung recruitment highlighted here with the dotted line. Pentachrome staining confirmed structural maintenance of main and lobar bronchi, as well as pseudostratified epithelium with mucus-containing goblet cells and ciliated airway epithelial cells, airway secretory glands, and airway smooth muscle myocytes. We confirmed the maintenance of small airway architecture by preparing airway casts, shown here. I'm not sure you can see it in detail in black and white. Primary respiratory epithelial cell phenotypes were preserved throughout the small airways. This was confirmed with immunostaining, including ciliated, neuroendocrine, club, and goblet cells. And then finally, in the distal lung, CFSE uptake confirmed cell viability throughout 24 hours of xeno cross-circ. And additionally, we observed type 1 cells amid normal distribution of epithelial tight junctions, and we confirmed type 2 cell viability and function by observing uptake of exogenous fluorescently labeled surfactant protein B, shown here. So now looking into the vascular compartment, at 24 hours, we also observed preservation of the pulmonary vascular tree, shown here on angiogram. We also observed retention of stereotypical elastic fiber organization in both arterial and venous vessel walls and normal endothelial cell morphology. We also demonstrated at the endpoint of xenogeneic cross-circulation that the lungs still retained vasoresponsiveness and bronchoresponsiveness. We showed this via delivery of phenylephrine, albuterol, and methacholine. Following the delivery, we observed expected changes in vascular and airway pressures respectively. And so what this really showed us was that not only do lungs on cross-circulation appear normal on histology and gross appearance, but also they really retain the ability to respond to exogenous stimuli in both the vasculature and in the airways. So to characterize the immunological effects of xenogeneic cross-circulation, we assessed retention of swine and human CD45 positive leukocytes in human lungs by staining for swine alpha-gal and human nuclear antigen. And after an initial increase, swine leukocytes in human lungs then decreased over 24 hours. Similar trends were seen with human and swine neutrophils and alveolar macrophages, which were initially elevated but then reduced by 24 hours. So kind of in conclusion, in terms of the path forward for cross-circulation, we really see two possible avenues for bringing this technology to patients. The first possibility would be to use the xenogeneic cross-circulation system that I described today to recover human donor organs with swine host prior to transplantation into the human recipient. So that would be more or less what I described today, taking those lungs off the circuit and transplanting them into a patient. On the other hand, we imagine a different scenario in which we would use human-to-human allogeneic cross-circulation. And in this configuration, a patient awaiting lung transplant who is supported on ECMO may be able to support the donor organ that they will eventually receive. So this would be achieved by kind of incorporating or splicing the donor lung directly into an already existing ECMO circuit, and allowing that donor lung to reach a level of function that is suitable for transplant, and then that patient would actually receive that as their lung transplant. Furthermore, we envision cross-circulation as a platform technology that could be used across various transplant and biomedical research applications. In particular, our group has recently begun to explore applications of cross-circulation in liver support. So perhaps with more to report on that at next year's meeting. And finally, our publication came this summer at a time when the news media was really, I think, ripe for some good news related to respiratory health, to say the least. So we were really excited that several mainstream media publications reported on our study, including the New York Times and some others listed here. So finally, this work is really the result of a huge team effort. I'm just the representative team member that had the good fortune of sharing the work with you all today. So we're extremely fortunate to work with many collaborators across departments and institutions, primarily Columbia and Vanderbilt University Medical Centers. We look forward to continuing this exciting work in the future. So thank you for your time, and I'm happy to take any questions.